Hello guys, welcome to our channel Mootechie. In this presentation, we'll explore the systematic study of bacteria, focusing on the three key processes that organize the microbial world. Meaning of bacterial systematics. This is defined as the scientific study of bacterial diversity, integrating classification, nomenclature, and identification. So, it's the holistic framework we use to understand, name, and categorize the vast world of bacteria. It organizes strains into natural groups reflecting phylogeny or evolutionary history. This means we group bacteria based on their shared ancestry, not just random similarities. Its scope covers prokaryotes in domain bacteria, distinct from archaea and eukarya. We are focusing specifically on true bacteria, separate from other fundamental life forms. Operational species concepts are now largely guided by specific genomic thresholds. Today, we often use precise DNA sequence comparisons to decide what constitutes a bacterial species. This systematics is built on polyphasic taxonomy, combining phenotypic and genotypic data. We look at everything from physical traits like cell shape to genetic information for a complete picture. Core components of systematics, the three pillars. Let's break down the three fundamental activities that make up bacterial systematics. First, classification. This is arranging organisms into taxonomic groups or taxa based on evolutionary similarity or relationships, for example, from domain down to species. Think of it as creating the family tree for bacteria, placing each organism in its proper branch. Second, nomenclature. This is the assignment of scientific names according to the rules of the ICNP, ensuring valid publication and legitimacy. This is the rulebook for naming, making sure every scientist worldwide uses the same officially recognized name for a bacterium. Third, identification. The practical process of determining that a new isolate belongs to one of the recognized taxa. When you find a new bacterium in the lab, this is the detective work you do to figure out what it is and where it belongs on that family tree. Now for tools and standards. We have type strains. These are living cultures serving as the permanent reference for a name, essential for comparative studies. They are the official representative specimen, the gold standard that all other bacteria of that name are compared against. And we rely on resources like Bergey's manual and LPSN, the list of prokaryotic names with standing in nomenclature. These are the essential encyclopedias and databases that every microbiologist consults for authoritative information. Classification systems. Let's look at how we've classified bacteria over time and how modern methods have revolutionized the field. Historically, phenotypic schemes relied on morphology, staining, and metabolic traits. Scientists used what they could see under a microscope and how the bacteria grew on different foods. Now, modern phylogenomic frameworks utilize core genes and whole genome sequencing, or WGS. We've moved to reading the organism's entire DNA blueprint to find the most accurate evolutionary relationships. The 16S rRNA gene is standard for higher ranks but often lacks species-level resolution. It's great for figuring out the bacterial family or genus, like identifying a surname, but sometimes not precise enough for the individual. Today, genomic metrics define species, ANI, or average nucleotide identity, at about 95 to 96% and dDDH, digital DNA-DNA hybridization, at about 70%. These are the hard numbers we calculate from genome data to objectively decide if two bacteria are the same species. The outputs of this work include ranked taxonomy and phylogenetic trees reflecting evolution. We end up with a detailed multi-level classification system and a visual tree that maps out their evolutionary history. On this slide, we also see a phylogenetic tree of bacteria. It's a visual representation of bacterial evolution showing major groups like proteobacteria, firmicutes, and actinobacteria branching off from a common ancestor. The labels you see, such as xylaxis and metagen, represent different bacterial groups or phyla. The tree illustrates how different bacteria are related based on genetic differences, with the scale bar indicating the rate of evolutionary change. Nomenclature rules, governed by the ICNP, the International Code of Nomenclature of Prokaryotes. These are the internationally agreed upon rules that bring order and consistency to bacterial names. Rule 1. Binomial names. Genus species in italics, authorship and year are optional. Just like Homo sapiens for humans, every bacterium gets a two-part Latinized name written in italics. Rule 2. Valid publication requires listing in the International Journal of Systematic and Evolutionary Microbiology and deposit of a type strain. You can't just name a new bug, you must publish it in the right journal and deposit the reference strain in a public culture collection. Rule 3. Priority and synonymy. The earliest validly published name generally prevails. The first correct name given to an organism usually sticks, preventing confusion from multiple names for the same thing. Rule 4. Infra-specific ranks, subspecies, pathovar, and serovar when relevant. For even finer detail, we can name variants within a species, like strains that cause specific diseases or have different surface proteins. The bottom of the slide lists various bacterial groups, illustrating the diversity of names and categories that fall under these naming rules. Identification methods. Now let's explore the practical tools we use in the lab to figure out exactly which bacterium we are working with. We start with phenotypic methods, gram stain, morphology, and biochemical panels like API or Vitek for classic analysis. These are the traditional hands-on tests that tell us about a bacterial structure and what chemicals it can use. MALDI-TOF MS provides rapid protein fingerprints compared against extensive reference libraries. This modern tool acts like a bacterial fingerprint scanner, identifying an organism in minutes based on its unique protein profile. Genotypic methods use PCR and sequencing of the 16S rRNA gene and other conserved housekeeping genes. Here we amplify and read specific sections of DNA, which is like checking the bacterial's genetic ID card. And with whole genome methods, we use high-resolution ANI-based matching and K-mirror classifier pipelines. This is the most powerful method, comparing the entire genome against databases for an extremely precise match. The workflow typically goes pure culture isolation, then a rapid screen like MALDI-TOF, followed by confirmatory genetic testing. We isolate the bacterium, get a quick idea of what it might be, and then use genetic tools to confirm its identity with certainty. The diagram shows common bacterial shapes like cocci, bacilli, and spirali with and without flagella, reminding us that morphology is often the first clue in identification. Methods in bacterial systematics. Let's dive deeper into the standard and modern techniques that power bacterial systematics. First, phenotypic profiling analyzes growth range, metabolism, and chemotaxonomy like fatty acid methyl ester analysis or cell wall composition. This builds a detailed profile of the bacterial's physical and chemical characteristics. Second, molecular typing uses methods like MLST, PFGE, and REPCR to differentiate strains with high resolution. These are specialized DNA-based techniques that can tell apart even very closely related bacterial strains, crucial for tracking infections. Third, phylogenetics involves reconstructing evolutionary history using concatenated core genes or specific markers like the 16S rRNA gene. This is how we build the family trees, using genetic data to map out how bacteria are related over time. Moving into the genomic era, we have comparative genomics. This defines species boundaries via ANI and dDDH metrics and analyzes pangenome diversity. By comparing whole genomes, we can draw clear lines between species and understand the total gene pool of a group. Finally, environmental methods employ metagenomics and metabarcoding to classify uncultured taxa directly from nature. This allows us to study the vast majority of bacteria that have never been grown in a lab by sequencing DNA straight from soil, water, or the human gut. The slide emphasizes that phylogenomics provides high-resolution insights, combining evolutionary biology with massive genomic datasets. Importance of bacterial systematics. Understanding how we classify and name bacteria isn't just academic. It has profound real-world impacts across several fields. In diagnostics, accurate identification ensures correct antibiotic therapy and supports antimicrobial stewardship. Knowing exactly which bug is causing an infection means doctors can prescribe the right drug the first time, saving lives and fighting resistance. For surveillance, it is vital for tracking outbreaks, monitoring antimicrobial resistance lineages, and identifying pathogen sources. It's the foundational tool for public health experts managing diseases and preventing their spread. It enables clear communication by providing stable nomenclature, enabling reproducible global research and data sharing. When every lab uses the same, correct name, scientists can reliably share and compare findings worldwide. In ecology and biogeography, it maps diversity to understand functional roles and environmental interactions. It helps us figure out what different bacteria are doing in an ecosystem, from the oceans to our own intestines. And in biotechnology, it facilitates the discovery of novel strains for industrial, pharmaceutical, and agricultural use. By systematically exploring bacterial diversity, we can find new producers of antibiotics, enzymes, or compounds for manufacturing. The colorful bacterial icons on the slide, representing groups like proteobacteria and firmicutes, visually symbolize this incredible diversity that systematics helps us navigate. Thank you. Subscribe for more microbiology content. We hope this clarified how scientists organize, name, and understand the invisible world of bacteria. If any questions popped up while watching, please share them below. We'd love to keep the conversation going. Happy learning!